Today we're talking about Chapter 11, Sections 3 and 4, and this is going to focus on other ways that populations can evolve that are not natural selection. Before we get into those other ways of populations evolving, we need to define some really important terminology. The first is gene flow. Gene flow means that there is movement of alleles between populations. So if individuals leave one population and they join a new population and start reproducing within that new population, that is introducing new alleles into that population that were not previously there. When you have a lot of gene flow between neighboring populations, that means those populations are going to stay pretty genetically similar. For example, people that live in the United States and live in Canada, there's a fair amount of gene flow between those two countries because we share a border and people can pass between those two countries. They can live in either country. They can find a mate in either country. And so genetically speaking, people in the United States are not really genetically all that different from the people who live in Canada. Now, if you have a low amount of gene flow, then that is going to increase the chance that the two populations are going to evolve into different species. That's probably not going to happen anytime soon in the world population, just because we are such a mobile society and a global society these days. People are able to travel all over the world. But if you think of other organisms that don't travel all over the world, maybe they're isolated on a certain island, that isolation and the lack of gene flow with other populations is what keeps them likely isolated reproductively and able to become a different species. Our next term that we need to discuss is genetic drift. Genetic drift is a general term that describes how allele frequencies can change just due to a random event. It could be as simple as some members of a population just happen to reproduce more often and have more offspring than others, and so those genes become more common, or the alleles for those genes become more common in the population. It could also be caused by a certain event, like what you see here, a bottleneck. We call this the bottleneck effect. A bottleneck is some kind of event, like a natural disaster, or if there's a disease that wipes out most of a population, we could have overhunting or overfishing in some natural populations. But if we take a large population and we reduce it in size so that only a few individuals remain, that surviving population might look really different genetically compared to the original. In this example, we have completely lost the red beads when we dump them from a large container into the small container. And this is why it's called the bottleneck effect, because if you were to think of it in terms of beads in a bottle and you were to dump out whatever beads are closest to the top, you could completely eliminate one or more alleles in your surviving population. The other main example of genetic drift is similar in effect, but the way that it happens is different. This is called the founder effect. And instead of killing off most of your population, in the founder effect, you have new individuals going and establishing a new population somewhere else. So in this example here, we have a population of flowers. They're all three different colors. But if a bird is taking the seeds or taking the flowers into a new area and producing a new colony from them, if it just so happens they only took the yellow and blue, now we only have yellow and blue flowers here instead of all three colors that we had at the beginning. So the end result is the same, it's just a different way to get to that end result. Both of these are examples of genetic drift. They are random changes in allele frequency because there wasn't any kind of selective force that made that orange color flower disappear. It was just random. Whatever color the bird had in its beak, those are the colors that made the new population. So genetic drift, which includes bottleneck and founder effect, those are both random changes in allele frequency. All right, another way that a population can change is called sexual selection as opposed to natural selection. Sexual selection is when an individual chooses who it is going to mate with. And if there's a pattern in that choice, if the females are always choosing males that look a certain way, then that's going to produce future generations of males that inherit those traits and tend to look the same way. This happens very often in species. This is what creates this bird's tail here. This is a widow bird here. It has a mating dance where it basically jumps off of the grass and flaps up in the air and it shows off its huge tail. Well, the female widow birds will only choose to mate with the males that have the longest tails. So even though this tail allows the male to get a mate, we don't really consider that natural selection because it's not giving a survival advantage. We're not talking about survival of the fittest here, because having a long tail actually makes that bird less likely to survive. But having a long tail makes it much more likely to mate. And remember, biological fitness is all about mating. Even if you have a trait that might make you die earlier, if you reproduce and pass down that trait before you die, it is going to become more common in the population. So we see that a lot throughout the animal kingdom in the tail feathers of the peacock that are very large and showy. They don't help that peacock survive at all. They are just there to get a mate. In fact, they are in the way when it comes to that male peacock's survival. We see aggressive behavior. We see things like large horns, large size, fighting over females. That can decrease the lifespan of a male, but the fact that he will do that and show that aggression is attractive to a female, and that's why that behavior is continuing. It's being selected for in the population. So in 11.4, we're going to build on those ideas of different ways that populations might have their allele frequencies changed. And we're going to apply a set of equations called the Hardy-Weinberg equations and use them to predict what the frequencies of the different phenotypes and genotypes of our population are going to be if it is not evolving. So that is the number one thing to remember about Hardy-Weinberg. These equations describe populations that are not evolving. We call that Hardy-Weinberg equilibrium, is when the populations are not evolving. And biologists really use this as a point of comparison. They can measure allele frequencies. They can see if those allele frequencies are changing. If everything is staying the same and it's remaining in equilibrium, then they say that the population is not evolving. Now, in order for the Hardy-Weinberg equations to be accurate, this is a mathematical model, so we are looking at a certain set of assumptions. These 5 conditions that you see listed here, they must all be met for Hardy-Weinberg equilibrium to occur. First condition is we have to have a large population because that is going to cut down on the chance of genetic drift occurring. Remember, we talked about genetic drift earlier in this presentation. We can't have allele frequencies changing due to random events and have a population still be in equilibrium. So the way you get around that is you have a very large populations, meaning thousands or millions in your population. Second condition is there can't be any individuals leaving or emigrating. You can't have anyone coming in or immigrating. So essentially, another way of saying that is you cannot have gene flow in your population. Remember, we are looking at assumptions that a population is going to be remaining stable and not evolving. So you don't want gene flow with another population that can change your allele frequencies. Third condition, no mutation. Now, is this really something that we can control? No, we know that mutations happen. So it's very difficult to make an argument that a natural population is going to have no mutations. But that is one of our assumptions for this mathematical model to work, is that we can't have new alleles being added to our gene pool. The fourth assumption is that the mating is random. We can't have any sexual selection. And along with that, number 5, there can't be any natural selection. We can't have one trait being beneficial over another, because then that trait is going to be selected for, and instead of having a population in equilibrium, we would then have a population that is not in Hardy-Weinberg equilibrium, and it would be evolving. So as we see here at the bottom of the screen, a real population is almost never going to meet all of these conditions. That doesn't mean that Hardy-Weinberg is useless. It just means that it describes a theoretical population that is kind of the ideal circumstance, kind of like in physics when you assume that there's a frictionless surface when you're trying to calculate velocity of something rolling down a ramp. This is kind of the same thing here. In order for the math to work exactly, we are making these 5 assumptions about our population. So what do we actually use Hardy-Weinberg for? It is useful for making predictions. We can predict the frequencies of different genotypes in our population if we know something about the allele frequencies that are there. So the way that this is used most common is if we know the frequency of our individuals that are homozygous recessive. Why is that important? Well, homozygous recessive means whatever phenotype they have, we can look at them and tell exactly what their genotype is. We can't do that for homozygous dominant because a homozygous dominant individual would look exactly the same as a heterozygous individual. But we do know that homozygous recessive has to have 2 recessive alleles in its genotype. So that's the key to solving all Hardy-Weinberg questions, is to determine homozygous recessive individuals first and then work backwards from that and solve the rest of the equation. So this is the equation that we're using most often. P squared is going to represent the frequency of our homozygous dominant individuals. 2pq is going to represent heterozygous, and q squared represents homozygous recessive. And if you are wondering where those numbers come from, we use P to represent a dominant allele, we use Q to represent a recessive allele. So basically, if we think about our gene pool for the population, if there are individuals in it that are heterozygous, it's possible to get 2 dominant alleles together, so P times P, the frequency of getting both of those at the same time, we multiply it, there's our P squared. We could get PQ or we can get QP, right? There's 2 PQs here. You can either get P from one parent and Q from the other, or you can get Q from the first parent and P from your second parent. So there's 2 ways to get the PQ term, and that's why we have a 2 in front of it. And then finally, Q squared, there's only one way you can be homozygous recessive. You have to know the probability of getting a Q allele from one parent and a Q allele from the other. You multiply them together, that's our Q squared. So in summary, the 5 factors that can lead to evolution, kind of pulling this all together. We can have genetic drift, which remember is a random process. This is not going to lead to new adaptations. It's just randomly some members of your population leave behind more offspring than others. We can have gene flow. That is also likely random, which individuals leave one population and go join a new one. That would be gene flow. Mutations are definitely random. They don't happen in set places in our DNA. They can happen wherever. We have natural selection that can lead to evolution. We've learned about that extensively. It's definitely not random, and it is the only one that actually produces adaptations or features that are beneficial for survival. And finally, we have sexual selection, which again is not random, but instead of survival, it's producing features that benefit in mating and reproduction. So that's going to be the end of sections 3 and 4. Hopefully you found this to be helpful, and if you have any questions, please ask them in class. We will be doing further practice with the Hardy-Weinberg equation, so if this video is not enough, don't worry, we have more practice that we're doing on that topic a little bit later.